Welcome, my dear students. We continue our lecture on functions of different cell structures. We have covered about cell membrane. Now we are going toward the cytoplasm structure. So if the nucleus is the office and the membrane is the security wall, the cytoplasm is the factory floor. It is the entire region between the nuclear envelope and the cell membrane where all the action happens. The cytoplasm is not just water. It's a thick jelly-like aqueous medium called cytosol. Think of it like a clear jelly that is packed with tools and materials. It is roughly 70 to 90% water, which provides a stable environment for organelles to float and for chemicals to move around. So floating within this jelly is a complex mixture of dissolved substances that the cell needs to survive, and it includes first of all salts and ions, okay, which are essential for electrical signaling and maintaining the cell's water balance. Then sugars and amino acids, which are the raw materials. Sugar provides the fuel, and amino acids are the bricks used to build proteins. And thirdly, proteins and enzymes. These are the functional tools which drive every single chemical change inside the cell. In eukaryotic cells, the cytoplasm is incredibly crowded. It is filled with specialized structures called organelles. You will find mitochondria producing energy, ribosomes building proteins, and vacuoles storing waste, all suspended in this jelly-like medium. And this crowding is actually very helpful as it brings the tools and the raw materials close together, so reactions can happen very quickly. So remember, the cytoplasm is the medium for life. It provides the space, water, and the chemicals necessary for the cells to function properly. And without this jelly-like medium, that is the cytoplasm, the cell's machinery would have nowhere to sit and nothing to work with. It is a very common mistake of the students to think of the cytoplasm as just empty space or filler between the organelles. In reality, it is the primary workspace where the cell lives, breathes, and does and performs its daily functions. The most important job of the cytoplasm is hosting metabolic activity. Almost all the cell's chemical reactions happening right here in this jelly. For example, did you know that the very first stage of respiration called glycolysis does not happen in the mitochondria? It happens directly in the cytoplasm. It is the prep station, okay, preparatory station where glucose is broken down before it is sent to the power plants for more energy. Secondly, the cytoplasm also provides support and shape. It creates an internal pressure that holds the organelles in their proper places so they do not just sink to the bottom of the cell. But it is not static. Remember, this is not static. It acts as a transport system. For example, in some plant cells, you can actually watch a process called cytoplasmic streaming, where the jelly flows in a circle around the vacuole like a conveyor belt, moving nutrients and signals to move corner of the cell. That is about transportation. And then the storage. When a cell has extra energy, it stores it right there in the jelly as tiny clumps. In plants, you will see starch grains. In animals, you will find glycogen granules or even tiny oil droplets. Think of these like snacks kept on the factory floor, ready to be used the moment the cell gets hungry for energy. So remember, the cytoplasm is the chemical lab, the conveyor belt, and the pantry of the cell. It is a dynamic environment, not static. It is a dynamic environment that keeps everything moving and reacting. To wrap up our look at the cytoplasm, let's go through this summary table. This covers the big three features that make the cytoplasm a functional workspace rather than just a bag of water. First is the water content, okay, at 70 to 90% water. The cytoplasm is the perfect solvent. In biology, almost every chemical reaction requires a liquid environment to happen. Because it is water-based, it allows salts, sugars, and gases to dissolve and react with one another. Without this water, the chemistry of life would simply grind to a halt. Next, let's talk about enzymes. Okay, which are floating in that water. These are your biological catalyst. We shall study that in detail under the section of under the chapter of enzymes as biological catalyst. Okay, the importance of these enzymes cannot be overstated. They speed up metabolic reactions millions of times over. Without enzymes to kickstart these changes, the chemical reactions needed to sustain your life would happen too slowly to keep you alive. The cytoplasm is essentially a high-speed chemistry lab. Finally, we have streaming, also known by its scientific name cyclosis. This is the physical flowing of the cytoplasm. It is incredibly important because it acts like an internal conveyor belt, constantly moving nutrients, oxygen, and even whole organelles to exactly where they are needed, where they are required to perform their functions. It ensures that no part of the cell is left starving or clogged with waste. So remember, water provides the medium, enzymes provide the speed, means they catalyze the reaction, they speed up the reactions, and streaming provides the support. Now we move outside the cell membrane to look at the cell wall. This is a rigid, non-living layer that acts as a protective shield. While we find it in fungi and bacteria also, today we are focusing on the version found in plants only, where its primary building block is a complex carbohydrate called cellulose. The structure of the cell wall is incredibly tough. It is made of cellulose fibers, which are long chains of glucose molecules bundled together into microfibrils. Okay, you can call that microfibrils. Think of these like the steel cables in a suspension bridge. They are woven together to create a material that is lightweight but strong enough to support the weight of a massive oak tree. A very important distinction for your exams, unlike the picky cell membrane, it is the cell wall that is freely permeable. Think of it like a chain link fence or a mesh net. It has a very open structure that allows water, mineral salts, and even large molecules to pass right through it without any restriction. It does not decide what enters and what leaves. It simply provides the structural frame of the cell to live in. Because the cell wall is non-living, it does not grow or change on its own like the membrane does. It is a fixed structure. But do not let that fool you. Okay, you should not be get confused. Without this rigid boundary, that is the cell wall, plants would not be able to handle the internal water pressure they need to stay upright. So remember, the cellulose provides the strength, and the mesh structure provides the access. It is the ultimate combination of protection and permeability. We have seen that the cell wall is made of tough cellulose. But what does that actually do for the plant? Its primary functions are rigidity, support, and protection. In many ways, the cell wall acts as the plant's external skeleton. First of all, regarding its function, okay, it provides a fixed shape. Because every individual cell is wrapped in a rigid box, they can be stacked like bricks. This allows the entire plant to grow tall toward the sunlight without collapsing. And beyond support, it is also a physical barrier. It protects the delicate internal parts of the cell from mechanical stress like wind or being stepped on, and acts as a first line of defense against pathogens like bacteria and fungi. Okay, then we are moving towards our second function, the second function of the cell wall, which is the perhaps most clever job of the cell wall. That is, it is preventing the cell from bursting. When a plant cell takes in water through osmosis, the vacuole swells up like a balloon. This pushes the cytoplasm hard against the cell wall. But in an animal cell, this pressure would cause the cell to pop, to burst. There is a process called lysis. But the plant cell wall is strong enough to push back. It resists that internal pressure, keeping the cell intact no matter how much water it takes in. If you talk about rigidity versus wilting, this internal pushing is called turgor pressure. When the cells are full of water and pushing against their walls, they are turgid or firm. This is what keeps a plant standing upright and healthy. If a plant loses water, that pressure drops. The cells go soft and the plant wilts. The cell wall is the frame that allows that pressure to do its job. So remember, the cell wall is the frame for growth, the armor against stress, and the safety valve that prevents the cell from bursting under pressure. It is the secret to plant's strength. The secret to plant's strength. Next, we look at the chloroplast. These are specialized membrane-bound organelles found primarily in the green parts of a plant, like the leaves. If the mitochondria are the power plants that use fuel, the chloroplasts are the factories that make the fuel. The most obvious feature of a chloroplast is its color. They are filled with a green pigment called chlorophyll. Think of chlorophyll as a specialized light trap. Its primary job is to absorb light energy, mainly from the sun. Without this specific pigment, a plant would be unable to capture the energy it needs to drive its chemical reactions. This is why plants turn brown or pale if they do not get enough nutrients to make chlorophyll. If you were to cut open a chloroplast, you would not just see a green soup. You would see stacks of disc-like membranes called thylakoids. These are, okay, here are thylakoids. These are disc-like membranes. These stacks are a brilliant piece of biological engineering. By stacking these discs, the chloroplast maximizes its surface area. Okay, they are stacked on each other to maximize the surface area. More surface area means more chlorophyll can be packed in, which means the cell can absorb much more sunlight in a shorter amount of time. And these thylakoids are where the first stage of energy capture happens. This energy collected here is then used to fuel the assembly of sugars. It is important to remember that chloroplasts are not found in every plant cell. You will not find them in the roots, for example, because there is no sunlight underground. They are only found where they can see the sun. So remember, the chlorophyll is the trap and the thylakoids are the structures which hold the trap. Together they turn a leaf into a massive solar array. So far we have seen the solar panels, but now let's look at the assembly line. Chloroplasts are the biological factories of the world. Their job is simple but miraculous. They take low energy ingredients from the environment and turn them into high energy food. The core function of the chloroplast is photosynthesis. Using the light energy captured by chlorophyll, the chloroplast drives a chemical reaction. It takes in carbon dioxide from the air and water from the roots. By rearranging these molecules, it produces glucose, which is a high energy sugar, and releases oxygen as a byproduct. This is the very reaction that keeps almost all life on Earth breathing and eating. In a photosynthesis chapter, you will study and discuss it in detail. Once the glucose is made, the plant has two choices. It can use it immediately for respiration in the mitochondria to get a quick energy boost, or if it has a surplus, it can join thousands of these glucose molecules together to form starch. You can actually see these starch grains under a microscope inside the chloroplast. It is like the plant is packing away energy boxes for a rainy day or a long night when the sun is not shining, means under harsh environment. In your exams, you will often be asked to name the inputs and outputs of this reaction of photosynthesis. Okay, remember, inputs are carbon dioxide and water, and the outputs are glucose and oxygen. And do not forget, this only happens in the presence of light. No light, no factory, no food. So remember, chloroplast is the processor. It takes raw materials and sunlight and turns them into fuel, that is glucose, and storage, that is starch. It is the foundation of the entire food chain. To wrap up our look at the chloroplast, let's go through this summary table. This covers the big three factors that determine how effectively a plant can feed itself: the pigment, the location, and the final product. First, we have the chlorophyll. As we have learned, this green pigment is the heart of the operation, heart of this function. Its importance cannot be overstated. It is the only molecule capable of absorbing light energy and starting the chemical chain reaction. Without chlorophyll, sunlight would just bounce off the leaf, and the plant would have no way to grab the energy it needs to survive. Next, look at where these chloroplasts are found. Means its location. The palisade and mesophyll cells. Okay, in a leaf, these cells are packed tightly together right near the upper surface. This is a brilliant piece of evolutionary design. By positioning the chloroplast as close to the leaf surface as possible, the plant ensures maximum light absorption. It's like putting solar panels on the roof of a house rather than in the basement. That if you want to generate solar energy, you will have to place your solar panels on the roof, not on the floor. Okay, this is the same phenomena in the leaves. Finally is the end product, which is the glucose. This is the ultimate goal of photosynthesis. Glucose provides the chemical energy that powers every single process in the plant, from growing new roots to producing flowers and fruits. But it is not just for the plant, because plants are at the base of the food chain. This glucose eventually provides the energy for every animal on earth, including us. So remember, chlorophyll is the trap, palisade cells are the best location for that trap, and glucose is the prize. That completes our study of the plant's solar panels, that is chloroplast. Uh, and next, we shall move towards our another organelle, that is large central vacuole. Okay, as an exam tip, if an exam if an examiner asks why leaves are thin and broad, you can link this slide that leaves are thin so light can reach the palisade cell easily, and broad to maximize the amount of chlorophyll exposed to the sun. A common question is why roots do not have chloroplast. If you remember the previous slide, okay, this is just a recap that since roots are underground in the dark, chloroplast would be waste of energy and space. That is the reason the underground parts, that is root, does not have chloroplast. To continue our tour of the organelles, we look at the large central vacuole. While animal cells sometimes have small temporary bubbles called vesicles, this massive permanent tank is a signature feature of a mature plant cell. It can often take up more than 80% of the cell's total volume. First of all, we shall discuss about cell sap. So inside this vacuole is a specialized fluid called cell sap. Do not confuse this with the cytoplasm. Cytoplasm is different and cell sap is different. Cell sap is a highly concentrated watery solution. It acts as a storage locker for the cell's most important spare parts. It contains sugars and amino acids, which are the cell's dissolved food reserves. It has mineral salts, which are essential for maintaining the cell's water balance. This large central vacuole also serves as a security vault. It can sequester or lock away waste products that might be harmful if they were floating around in the cytoplasm. By trapping them inside the vacuole, the rest of the cell stays clean and protected. In some plants, the vacuole even stores toxins or bitter chemicals to stop animals from eating the leaves. That is their survival behavior. If we talk about pigments and pollinators, in the cells of flowers, the vacuole contains pigments. These are the bright red, purple, and blue you see in petals. These colors are not just for show, they are designed to attract pollinators like bees and butterflies. So every time you see a colorful flower, you are actually looking at the contents of millions of tiny vacuoles. So remember, this cell sap is the solution and the vacuole is the tank. It stores food, hides waste, and provides the color which keep the plant's life cycle moving forward. We have seen what is inside the vacuole, but now let's look at how it acts as a high pressure hydraulic system. For a plant, the vacuole is not just a storage tank. It is the engine which provides structural support. Think of the vacuole like a balloon inside a cardboard box. When water enters the vacuole through osmosis, the balloon begins to swell. As it expands, it pushes the cytoplasm hard against the rigid cell wall. This outward force is called turgor pressure. Just like blowing air into a balloon inside a box makes the whole structure hard and sturdy, turgor pressure makes the plant cell firm and strong. When the pressure is high, we say the cell is turgid. For non-woody plants like flowers or grass, this is their primary means of support. They do not have wooden trunks like big trees or internal skeletons. They stay upright simply because every single cell is pumped full of water and pushing against its neighbors. But the question is, what happens when a plant gets thirsty? So when there is not enough water, the vacuoles shrink and pressure drops. The cells become flaccid, okay, flaccid or soft. Without that internal balloon pushing against the wall, the structure collapses and the plant wilts or droops. This is the plant's way of telling you it needs a drink to get its pressure back. So remember here that water intake leads to turgor, which leads to turgidity. Okay, water intake leads to turgor, which leads to turgidity. It is the secret to why plants can stand tall and reach for the sun. Here is the quick recap and the summary of vacuole. That it has three features: tonoplast, cell sap, and turgidity. Tonoplast, that is a vacuole's membrane. Okay, it regulates what enters us into the sap. Cell sap, this is the concentrated solution. It creates the osmotic gradient to pull water in. And turgidity, that is a state of being firm. It keeps the leaves spread out to catch sunlight. Remember here, a turgid cell is a healthy plant cell. It is the opposite of a plasmolized cell, where the vacuole has shrunk so much the membrane pulls away from the wall. So, if a question asks how a plant competes for light, mention that turgidity keeps the leaf canopy spread wide. Now we move to one of the most hardworking parts of the cell, that is the mitochondria. Whether it is a muscle cell in an athlete or a leaf cell in a tree, mitochondria are found in almost all eukaryotic cells. But what is their primary job? Their primary function is to act as the cell's energy converter, means they convert cellular energy. If you talk about their structure, the mitochondria has a very distinct internal architecture. It starts with a smooth outer membrane, which acts like a protective skin, controlling what enters the organelle. But the real magic happens inside at the inner membrane. Unlike the outer skin, this inner layer is highly folded into finger-like structures called cristae. But what is the function of this fold? You can also call it as power of folds. That is the surface area. That why it is so folded. In biology, folding almost always means one thing. Remember it: maximizing surface area. Okay, if you remember about thylakoid, which are sac-like structures, okay, they also increase the surface area, and here the folding is also for maximizing the surface area by having these cristae. Okay, in this picture you can look this at here. Okay, these are cristae, folded structures. So by having this cristae, the mitochondrion creates a massive amount of space for chemical reactions to occur. It's like having a giant laboratory table folded up inside a small box. The more table space you have, the more work you can get done at once. And finally, the very center of the mitochondria is filled with a fluid called the matrix. This is the matrix here, okay? This fluid, okay? This is the matrix. This space is packed with specialized enzymes which are essential for the process of respiration. It is here that the fuel from your food is processed to release energy which the cell needs to stay alive. So remember, the outer membrane protects, chemistry provides the workspace, and the matrix holds the chemical tools. And together they form the most efficient energy factory on the planet. So if an examiner asks why a specific cell has many mitochondria, the answer is always the cell has a high energy requirement for specific function. For example, the contraction of muscles or the active transport. That is the reason a few specific cells have more than one mitochondria. This link between structure and function is a core IGCSE objective and even level exams. So golden rule, always remember increased surface area is equal to increased rate of reaction. This is a universal rule in biology which applies to mitochondria, lungs and even intestines. Now let's look at the actual work happening inside those folds. We often say we breathe to get oxygen into our lungs, but the real breathing happens here at the chemical level. This process is called aerobic respiration. To keep the lights on, the mitochondria need two things: glucose from the food you eat and oxygen from the air you breathe. So the goal is to break down the chemical bonds in that glucose to release energy trapped inside. As the mitochondria burn this fuel, they release two byproducts, carbon dioxide and water. You can see this equation. This is glucose C6H12O6, and in the presence of oxygen molecules, they produce carbon dioxide, water molecule, and they release energy. This is the energy, adenosine triphosphate. It is the exhaust fumes from your cellular engines. But the energy is not just floating around loosely. The mitochondria capture it and pack it into a very special molecule called ATP, adenosine triphosphate. Think of ATP as the battery or the cache of the cell. The cell cannot use a sandwich to move muscles. Okay, they cannot move that muscle directly. It has to convert that sandwich into ATP first. Once you have ATP, means energy, the cell can spend it instantly to do work. But what kind of work? Everything. Every time you contract a muscle to walk, every time a neuron sends a signal in your brain, or every time your body builds a new protein to repair a cut, it is spending the ATP made by your mitochondria. And without this constant supply of molecular cash, the economy of your body would come to a complete standstill. So remember, the inputs are glucose and oxygen. The output is ATP, adenosine triphosphate. The byproducts are carbon dioxide and water, as it is shown here in this equation, which I explained you earlier. Okay, so the mitochondria are the reason you eat and breathe. Here is a common mistake which many students make, that they do not say mitochondria create energy. Okay, in fact they release or convert energy from glucose. To wrap up our study of the mitochondria, let's look a quick recap at the summary table. We have seen how they work, but now we need to understand why some cells have more engines than others. First of all, the chemistry. As a reminder, these are the deep folds of the inner membrane. Their importance is purely mathematical. They maximize the surface area. More surface area means more space for the electron transport chain and other reactions to happen. The more table space you have in your factory, for example, then the more ATP you can churn out every second. Next is the matrix, that central fluid space. This is where the Krebs cycle, we still did not study about Krebs cycle, but definitely in future you will study about that. So this is where the Krebs cycle takes place, which is the key part of respiration. Okay, the matrix is packed with specialized enzymes which act like the workers on the assembly line. They take the raw materials from your food and start the process of stripping away the energy to be packed into ATP. And finally, notice that the number of mitochondria is not the same in every cell. It varies based on how much work the cell does. For example, a skin cell might only have a few hundred, but a muscle cell or a liver cell, which are chemically very active, will have thousands of mitochondria. If a cell needs to move, pump, or build constantly, it needs more power plants to keep up with the demand for ATP. So remember, history provide the space, matrix provides the tools, and the number of mitochondria means their quantity that provides the scale. That concludes our deep dive into the powerhouse of the cell, that is mitochondria. Now we have covered all the major organelles. In our next lesson, next slide, we will look at ribosomes and how the cell actually builds the proteins we have been talking about. Uh, before moving to next slide, here is an exam tip for you, that we sometimes have a very common question, ask why a muscle cell has more mitochondria than a fat cell. The answer is always muscle cells require more energy for contraction. Therefore, they need more mitochondria to produce ATP through aerobic respiration. Dear students, if the nucleus is the architect with the blueprints, the ribosomes are the construction crew. These are tiny non-membrane bound structures found in every single living cell on earth, from the simplest bacteria to the most complex human cells. The primary job of a ribosome is protein synthesis. They take the genetic code sent from the nucleus and translate it into physical three-dimensional proteins. Whether it is the hemoglobin in your blood, the keratin in your hair, or the enzymes in your stomach, a ribosome built it. There are two primary locations of ribosomes. In eukaryotic cells, you will find ribosomes in two different workstations, depending on where the protein is headed. For example, the first one is free ribosomes. These float freely in the cytoplasm. They usually make proteins which stay at home to be used inside the cell, like the enzymes which break down sugar in the cytosol. The next one are bound ribosomes. These are attracted to the rough endoplasmic reticulum, giving it that signature sandpaper look. These ribosomes are the export specialists. You should also you should also know that proteins made by the ribosomes on the rough endoplasmic reticulum, we also write that as RER. Okay, so the proteins which are made by the ribosomes on the rough endoplasmic reticulum, those are destined for bigger things. They have bigger tasks. They are usually packaged up to be exported out of the cell. Like for example, think of hormones like insulin or digestive enzymes like amylase. Okay, these are examples. They can also be built directly into the cell membrane to act as those channels or receptors we studied earlier. So remember here, as a summary note, that free ribosomes make proteins for internal use, while bound ribosomes make proteins for export. They are the tiny engines that turn genetic information into living matter. As an exam tip, students often forget that ribosomes are found in prokaryotes also. It is one of the few structures they share with us. We have seen where ribosomes live, but now let's look at the actual assembly line in action. This is the moment where the cell's instructions become physical reality. This process is called protein synthesis. The nucleus is the boss, but it never leaves its office. Instead, it sends out a messenger molecule, which is called as messenger RNA. We write it like this way, where M is smaller and RNA is in capital letter. This messenger RNA carries the genetic blueprints directly to the ribosome, and the ribosome then locks onto this strand and begins to read the code one letter at one time. As the ribosome reads the code, it calls for specific building blocks called amino acids, okay, called amino acids. It links these together in a very precise order to form a long flexible string called a polypeptide chain, okay, this polypeptide chain. Think of this like a long string of beads. The order of the beads is determined entirely by the instructions sent from the nucleus. And then a long string of amino acids is not a protein yet. Okay, so to become functional, that polypeptide chain must fold into a complex 3D shape. If it folds one way, it might become an antibody to fight disease. If it folds another, it becomes a structural fiber for your muscle, or it might become an enzyme to digest your dinner. The shape is the function. Okay, form is the function, shape is the function. If it does not fold correctly, the protein would not work. So remember, the messenger RNA is a blueprint, the amino acids are the bricks, and the ribosome is the builder. And without this process, your DNA would just be book that no one ever read. As an exam tip, if a question asks for the definition of a ribosome, always include this phrase, the site of protein synthesis. Okay, that is the golden phrase for getting high marks, for getting good marks. Okay. With each slide, with each lecture, I am giving you a few exam tips which will be very convenient for you to prepare your exams. So to finish our look at ribosomes, let's look at their technical specification. Even though they are the smallest structures as we have discussed, the unique design is what makes them so efficient at building the proteins of life. First of all, the size. Ribosomes are incredibly small, only about 20 to 30 nanometers across. To put that in perspective, you could fit thousands of them inside a single mitochondrion. Because they are so tiny, they are invisible under a standard light microscope. You need the incredible power of an electron microscope to actually see these dots scattered throughout the cell. Next is the structure. Ribosome is not one solid piece. It is made of two parts, large subunit and a small subunit. Think of them like the two halves of a burger. ठीक है, तो two halves of a burger bun. When it is time to work, they clamp together onto the messenger RNA strand, holding the genetic code steady so it can be read accurately. Once a protein is finished, the two pieces simply drift apart again. Finally, we have diversity. Not all ribosomes are the same. Prokaryotes like bacteria have 70S ribosomes, okay, which we have discussed earlier in this chapter, okay, these are slightly smaller and simpler. But eukaryotes like plants and animals, they have 80S ribosomes, which are larger and more complex. This difference is actually life-saving. Many antibiotics work by specifically targeting the 70S ribosomes in bacteria to stop them from making proteins while leaving our 80S ribosome completely unharmed. So remember, size keeps them numerous, subunit keeps them functional, and the diversity is a key feature in medicine. That concludes our study for the cells construction crew. And in our next slide, we'll see how these ribosomes join forces with the rough endoplasmic reticulum. So the highway of the cell, as that is the rough endoplasmic reticulum. Now that as we have met the ribosomes, let's look briefly at the main workstation, that is the rough endoplasmic reticulum (RER). This is an extensive system of membranes which stretches out from the nucleus like a complex highway system filling much of the cytoplasm. But why we call it rough? The name rough comes from its appearance under an electron microscope. You can also see that here. It looks very different, okay, from a smooth surface. This is not a smooth surface. Its surface is studded with thousands of tiny ribosomes, giving it a granular sandpaper-like texture. These ribosomes are docked there because the proteins they are building need to be processed and shipped immediately. And structurally, By structure, the rough endoplasmic reticulum is made of a network of flattened membrane-bound sacs called cyst sorry called cisternae. Okay, think of these like a series of interconnected hallways. These are here. Okay, these are interconnected hallways. Think of the so inside these hallways, the proteins built by the ribosomes, which can be folded, modified, and safely transported away from the busy soup of the cytoplasm. One of the most important features of the rough endoplasmic reticulum is that it continues with the nuclear envelope. There is no gap between the nucleus and the RER, means rough endoplasmic reticulum. This physical connection is a brilliant design for efficiency. It allows the messenger RNA instructions to travel directly from the nucleus to the ribosomes on the RER with zero delay. So remember, the ribosomes provide the rough texture, the cisternae provide the internal space, and the connection to the nucleus ensures a high-speed production line. It is the cell's premier site for building and moving complex proteins. In the free ribosomes are like home cooks. The rough endoplasmic reticulum is a massive industrial factory. It does not just build proteins; it processes, modifies, and ships them to their final destinations. So let's look at how this assembly line actually works. As the attached ribosomes build protein chains, they feed them directly into the internal space of the rough endoplasmic reticulum, which is called as the lumen. Lumen. Okay. Once inside this protected hallway, the protein is away from the busy cytoplasm. Here it begins to fold into its specific 3D shape. The RER then acts as a conveyor belt, transporting these growing proteins through its network of sacs toward the next station. But raw protein often is not ready for work yet. The RER helps to modify these molecules. A common example is adding carbohydrate chains to a protein to create glycoprotein. Like in this picture, this is glycoprotein. Okay, think of this like adding a mailing label or a protective coating to a product before it leaves the factory. So these modifications tell the cell exactly where that protein needs to go and what its job will be. Finally, how does the protein leave the factory? The rough endoplasmic reticulum uses a clever budding process. A small piece of the RER membrane pinches off to form a tiny bubble called a transport vesicle. This vesicle acts like a shipping container, carrying the newly made proteins safely to the Golgi apparatus for final packaging. It is a seamless transition from production to delivery. So if a question asks about the pathway of a secreted protein, the order is always from ribosome to rough endoplasmic reticulum, okay, from ribosome to rough endoplasmic reticulum, then to vesicle, and then to Golgi. This is a very common flowchart style question. This is the last slide of today's lecture. So we are wrapping up our look at the RER. So let's go through its summary table quickly. As we have seen that how it possesses proteins, but now we need to understand why its location and density are so critical to your health. First of all is the appearance. As we have seen that rough endoplasmic reticulum gets its rough name because it is studded with the thousands of attached ribosomes. This is not just for show, but having the ribosomes physically docked on the membrane. It allows the protein chains to be injected directly into the transport system as they are being built. So it is the ultimate just-in-time manufacturing process. Next is the location. It's always found huddled near the nucleus. This is all about speed and access. Since the instructions for building proteins come from the nucleus, the rough endoplasmic reticulum sits right at the exit door. This ensures the assembly lines get their orders immediately with no time wasted traveling across the cell. Finally, notice the density. Not every cell has the same amount of RER. It is most abundant in secretory cells. Okay, for example, take the pancreas. For example, its job is to churn out massive amounts of the hormone insulin and digestive enzymes. Because these are all proteins destined for export, pancreatic cells are packed with rough endoplasmic reticulum to keep up with the high production demand. So remember, appearance tells you it is a factory, location gives it speed, and density tells you how much work that specific cell is doing for the rest of the body. So here we close our lecture. Okay, no doubt it is a bit longer than the earlier lectures, but and definitely these are quite in detail. But I'm very sure that if you attended this lecture with your complete attention and complete focus, and you noted down the your notes, you will not need to consult any other source. This definitely you will have to consult with the books, but by watching these videos, these are also sufficient for the preparation of your exams. So finally, is definitely an exam tip for you, that if you are asked about to identify a cell that produces a lot of enzymes, look for the one with the most rough endoplasmic reticulum. Okay, the pancreas, for example, that is the gold standard for producing a lot of enzymes. Okay, it means that has the most rough endoplasmic reticulum. So say stay subscribed to this channel. Okay, and also share with your colleagues, with your fellows, with your peers, and definitely we shall meet in our next video. Okay, and then we shall, in the next video we will cover completely this chapter. We shall conclude this chapter, and at the end of the video we shall have also practice that what we have learned in this chapter so far. Okay, so see you in next video. Till then, goodbye.